It is one of the most fascinating and, frankly, terrifying paradoxes in modern biology. I mean, think about it. Why is it that horseshoe bats act as these natural reservoirs for incredibly dangerous coronaviruses, just carrying them around without so much as a sniffle, right?
Right.
But then humans infected with those exact same viral families suffer severe and often fatal disease.
It really is the ultimate biological mystery because, you have these bats flying around perfectly healthy, essentially acting as flying viral storage facilities.
Yeah.
Meanwhile, the moment one of those viruses makes the jump to us, well, we are dealing with a global pandemic. The divergence in how our immune systems handle the exact same pathogen is just staggering.
Which is why we are taking you on a microscopic journey today to answer that exact question. We are looking at a highly revealing 2026 paper from the journal Cell Host and Microbe, published by Batra and colleagues.
It's a fantastic paper.
It really is. And the mission of this deep dive is to basically unpack their massive molecular map. We want to find the exact biological switches that allow a virus to jump species, evade our immune systems, and just, wreak havoc.
And the reason we are only just now getting to the bottom of this dynamic is purely logistical. For years, virologists and immunologists have been trying to study this reservoir-host relationship, but they lacked a fundamental tool.
Which was working bat cell line, right?
Exactly. They needed bat cell lines that could actually be robustly infected with SARS-CoV-2 in a laboratory setting. I mean, you simply cannot study the bat's intracellular defenses if you can't get the virus to sustainably interact with bat cells in a petri dish.
So before the researchers could even begin to map out the host-virus interactions, they had to build the biological stage itself.
Like they had to start from scratch.
And they created a brand new, immortalized lung fibroblast cell line from a greater horseshoe bat. Specifically, they used lung tissue from a Rhinolophus firmaquinum bat that was captured in Spain back in 2020. And they dubbed these RFAS cells.
The creation of that cell line is a feat in itself, honestly, because primary cells taken directly from an animal will naturally hit what's called the Hayflick limit.
Okay. For those who might not remember, that's basically a cell's division expiration date, right?
Exactly. They divide a certain number of times, they age, and then they just die off. So to make them immortal, the researchers introduced the SV40 large T antigen.
Which is essentially using it as a biological pause button on aging.
That's a really good way to look at it. The SV40 antigen binds to key tumor suppressor proteins in the cell, specifically like P53 and ARP.
And those normally tell the cell to stop dividing.
Right. By blocking those suppressors, it takes the brakes off the cell cycle, it forces the bat lung cells into a state of continuous division, and that gave the researchers a stable, reproducible canvas.
But having the canvas is only half the battle.
Yeah.
Because bat cells don't naturally express the specific entry receptors that human-adapted SARS-CoV-2 looks for.
No, they don't.
So the team actually engineered these bat cells to express human ACE2 and TMPRSS2. Now, hold on. Let's unpack this for a second.
Sure.
If they engineered the bat cells to have the exact human ACE2 doorway, and then, as the paper notes, they still had to manually mutate the virus's nucleocapsid protein just to force it to replicate inside the cell, doesn't that sort of compromise the whole study?
How do you mean?
Well, are we even looking at wild-type biology anymore, or is this just a lab-created artifact at this point?
It's a very fair critique, and it's actually one the authors explicitly address. You have to consider what that barrier truly represents. The researchers gave the bat cell the human ACE2 receptor. So the wild-type virus used that doorway, got inside the cell, but then it hit a complete intracellular brick wall.
Oh, so it got in, but it couldn't actually do anything.
Precisely. It couldn't replicate. By identifying the exact key required to overcome that internal wall, which is a single mutation in the viral nucleocapsid known as NK80T, they proved a vital concept.
Meaning that cross-species transmission involves host-specific barriers that are way beyond just the ACE2 receptor on the surface.
Exactly that. The NK80T mutation essentially acts as a forged biometric pass for the cell's interior.
I love that visual. So you have the key to the front door, but the interior doors of the house are all biometric.
Right. And without that specific amino acid change in the protein shell protecting the viral RNA, the bat cell's internal environment remained completely hostile to the human-adapted virus. This proved that zoonotic spillover requires complex, multi-layered adaptations. It's not just a simple key fitting into a surface lock.
Wow. Okay, let me pause to make sure we're tracking the sequence of events here, because the setup alone is incredibly elaborate. To even study this, they couldn't just use standard animal models.
No, not at all.
They had to immortalize a primary bat lung cell, bypass the surface receptors by adding human ACE2 and then overcome an internal biometric lock, that nucleocapsid mutation, just to get the virus to sustainably replicate.
Yes, that was just the prep work.
Right. And only after building that stage could they actually see what the virus was doing. Which brings us to the methodology. How do you actually map the behavior of a virus inside a cell?
Well, they turned to a technique called affinity purification mass spectrometry, or APMS.
APMS. Now that sounds dense, but the best way to picture it is like a microscopic fishing trip.
That's a great analogy.
Imagine you take a single viral protein, let's say the viral spike protein or the nucleocapsid we just mentioned, and you use it as your fishing lure. To make it retrievable, the researchers attach a microscopic molecular tag to it, specifically a 2X strep tag. Then they drop this tagged viral lure into the cellular soup of the infected bat cell.
And inside that incredibly crowded cellular soup, the viral protein just naturally binds to various host proteins. I mean, it binds to the exact cellular machinery it's actively trying to hijack.
Right. Then they physically pull the fishing lure back out using that 2X strep tag.
Right, they yank it out.
And whatever host proteins are stuck to the lure get pulled out with it. Then they run that entire catch through a mass spectrometer.
Which acts like a hyper-precise scale, basically.
Exactly. It identifies the exact molecular weight and identity of every single host protein that bit the hook.
And the scale of this comparative mapping is what makes this paper a landmark. They didn't just drop these lures into bat cells infected with SARS-CoV-2. They mapped a four-way interactome.
A four-way interactome.
Yeah, they looked at SARS-CoV-2, and they also looked at its closest known bat relative, a virus called RATG13.
Which shares over 96% of its genome with the human virus, right?
Right. So they ran the fishing trip with the human virus in human cells, the human virus in bat cells, the bat virus in human cells, and the bat virus in bat cells.
That is exhaustive. What were the quantitative differences?
They were stark. When looking at SARS-CoV-2 inside the newly created bat cells, the mass spectrometer identified 72 high-confidence protein-at-protein interactions.
Okay, 72.
Meanwhile, in human cells, the interactions numbered in the hundreds.
That discrepancy is fascinating. It implies the human-adapted virus is just running rampant, grabbing onto everything it can in the human cell. Whereas in the bat cell, its network is much tighter.
Yeah, it's perhaps far more restricted by the host environment.
That makes total sense. So before we get into the crazy molecular switches they found, let's do a quick bullet point recap for you listening, because we've covered a lot of ground in these first few minutes.
Good idea.
First, we built a custom bat cell line because we needed a stable canvas. Second, we found that the virus needed a specific nucleocapsid mutation, a forged biometric pass, just to replicate inside it. And third, we used molecular fishing, or APMS, to map every single protein the virus touches in bats versus humans.
That's the foundation, perfectly summarized.
So with this giant comparative map in hand, what did it actually reveal? Let's start with what the virus does identically in both species.
Well, first off, they found that regardless of whether the virus was in a human or a bat, it targeted the exact same essential cellular infrastructure.
We're talking about fundamental survival mechanisms here, right? Like RNA processing, translating viral proteins, moving those components around the cell.
Yes, the core tactics are shared. A prime example is the viral nucleocapsid protein, the N protein. The mass spec showed that in both humans and bats, the N protein acts as a sort of saboteur. It specifically binds to host proteins called G3BP1 and G3BP2.
G3BP1 and 2, those are the proteins responsible for forming stress granules, right? Like the cell's early warning system.
They are. When a cell detects viral RNA, it normally halts its own protein translation. It forms these dense clumps of RNA and proteins, which we call stress granules.
Sort of locking down the facility.
Exactly. These granules help organize an antiviral response and trigger the production of interferons. By having its N protein bind to G3BP1 and 2, the virus essentially dismantles the cell's early warning alarm, and it does this in both species.
Okay, but, and I think this is the core question, if the virus uses the exact same tactics, like dismantling the same alarms and targeting the same infrastructure in both humans and bats, why do you and I end up in the ICU with severe lung inflammation, while the bat just handles the infection seamlessly?
Well, the answer lies in the concept of evolutionary arms races, and this is specifically highlighted by a host protein called ZAP.
ZAP. That stands for zinc finger antiviral protein, right?
Yes. The fishing trip caused ZAP interacting with the virus in both species.
And ZAP's primary job is to bind to viral RNA and recruit cellular machinery to just degrade it.
That is its function, yes.
Huh.
But here's the catch. While the virus targets ZAP in both species, the ZAP protein itself has a very low sequence identity when you compare humans and bats.
Oh, interesting.
Because viruses constantly try to evade it, ZAP is forced to evolve incredibly fast.
So even when the virus grabs the exact same tool in a human versus a bat, the tool itself is physically shaped differently.
Right, due to millions of years of distinct evolutionary pressure.
But how does that structural difference actually change the biological outcome? I mean, how does it physically lead to a different result for the bat versus the human?
It completely changes the downstream signaling cascade. See, in humans, when our ZAP protein binds to the virus, it doesn't just quietly degrade the RNA. It interacts with other pathways that essentially scream at the innate immune system to launch a massive, scorched earth inflammatory response.
Oh, wow. The notorious cytokine storm.
Exactly.
So it's our own hyperactive immune response causing the severe lung damage, not just the virus replicating.
Yes, the host response is what kills you. But in bats, their ZAP protein has been calibrated by millions of years of coevolution with coronaviruses.
So it's used to them.
Exactly. It still binds the viral RNA and targets it for degradation, but it lacks the same volatile structural domains that trigger that massive inflammatory cascade in humans. It basically executes a quiet, surgical degradation of the RNA.
I see.
The viral load is kept in check, but the bat's lungs aren't destroyed by its own immune system.
So the bat fights back with a scalpel, while the human immune system just swings a sledgehammer.
That is a perfect way to describe it.
Okay, so that explains how host proteins evolve differently to handle the exact same viral tactics. But the paper highlighted something even more specific than different host responses. They found an instance where the virus itself completely changes its behavior depending on the host.
You are talking about ORF9b.
I am. Now, I know ORF9b is a notorious troublemaker in human infections. It's an accessory protein that essentially tackles our mitochondrial alarms. Does it try the exact same trick in the bat cells?
This is where the paper uncovers a literal molecular switch. And it is wild. In human cells, we know SARS-CoV-2's ORF9b heavily targets a host protein called TOM70.
TOM70, let's give a visual for that. We usually think of mitochondria just for energy generation, the powerhouse of the cell. But here, they act as the central hubs for our innate immune signaling.
Right, and TOM70 is a mitochondrial import receptor that helps activate MAVS. That's the mitochondrial antiviral signaling protein.
And MAVS is basically the signaling hub that tells the nucleus, hey, we are under attack, produce interferons.
Exactly. So by binding to TOM70, ORF9b effectively blinds MAVS. The interferons are never produced, allowing the virus to replicate wildly under a cloak of invisibility.
That is the human mechanism.
Yeah.
But the mass spec fishing trip in the bat cells revealed something totally different, didn't it?
It was staggering. In the bat cells, the ORF9b protein from the bat virus RATG13, or even from a mutated SARS-CoV-2, doesn't bind to TOM70 at all.
Wait, really? It ignores it?
Totally ignores it. Instead, it binds to a totally different protein called mTARS2.
mTARS2. And that is a bat-enriched restriction factor, meaning its primary biological function is to actively interfere with and slow down viral replication.
Yes. So instead of tackling the gatekeepers to shut down alarms like it does in humans, the viral protein in bats binds to a mechanism that actively limits its own spread.
That is wild.
Yeah.
ORF9b is acting as a literal molecular double agent. But what actually causes it to switch targets? How does the virus mechanically know to bind TOM70 in humans to cause chaos, and then bind mTARS2 in bats to maintain a stalemate?
Well, it comes down to the smallest possible biological change, a single amino acid substitution.
Just one.
Just one. The researchers pinpointed this switch to residue 72 on the ORF9b protein. In the highly pathogenic human SARS-CoV-2 virus, residue 72 is an amino acid called threonine. We call it T72.
Okay, T72 for humans.
But in the bat virus, RTG13, that exact same spot is occupied by an isoleucine or I72.
One single amino acid out of the entire protein chain acts as a switch.
One amino acid. But that single variation completely reshapes the physical interaction surface of the protein.
How so?
When it has the human configuration, T72, the viral protein surface naturally locks into the structural groove of human TOM70. It maximizes immune evasion, resulting in the high viral loads and the severe disease we see in humans.
But when it has the bat configuration, I72.
Its shape changes just enough that it bypasses TOM70 completely. It instead locks onto the bat's mTARS2 restriction factor. And by making that specific connection, the virus intentionally keeps itself in check.
It leads to a peaceful equilibrium.
Exactly. The bat stays completely asymptomatic. It is an incredibly delicate, natural balancing act.
That is an elegant piece of biology. Let's do our second quick recap here, right around the 2/3 mark of our deep dive.
Sure.
So we learned that some viral tactics are universal, but host proteins like ZAP evolve to fight back differently, the scalpel versus the sledgehammer. We also learned that ORF9b is a viral double agent. And finally, residue 72 on that protein flips a switch. In humans, it binds TOM70 for maximum damage, but in bats, it binds mTARS2 for a peaceful stalemate.
You've got it. And understanding these minimal sequence variations, like the switch at residue 72, is what moves this from, just an interesting biological puzzle to a crucial tool for global public health.
Because it gives us a genetic blueprint.
Precisely. This is how we predict zoonotic spillover.
So when field virologists are out surveying bat populations, sequencing newly discovered coronaviruses, they aren't just guessing about the danger anymore.
No, they have a map. If they sequence a novel bat virus and see that it naturally possesses the T72 configuration on its ORF9b protein, or really any configuration that shows a high affinity for binding human TOM70, they know immediately that this specific virus is already primed for immune evasion in humans.
That is chilling.
It is. It requires very few, if any, additional mutations to become highly pathogenic to us. It acts as a targeted early warning system.
And beyond just prediction, mapping these exact interactions gives us glaring targets for future therapeutics, right?
Absolutely.
Because if we know unequivocally that severe human disease relies heavily on the virus using ORF9b to bind to TOM70, then breaking that specific interaction becomes the prime directive for drug development.
That is the clinical endgame. If pharmacologists can design a small molecule inhibitor that chemically blocks ORF9b from docking with TOM70 in a human patient, well, you effectively strip the virus of its invisibility cloak.
And then our immune system can actually do its job properly.
Right. The human immune system would recognize the threat early, trigger the proper interferon response, and clear the infection before that devastating cytokine storm ever has a chance to develop.
Which brings up a really provocative thought to leave you with today. We have spent this deep dive exploring how the virus adapts to its host environment. But think about the raw mechanics of that molecular switch we just talked about. If a single amino acid mutation at residue 72 acts as a binary switch that turns a peaceful, self-limiting bat virus into a deadly, immune-evading human pathogen, could we actively use that mechanism against it?
Oh, that's an interesting question.
Right. Could we design a therapeutic drug that chemically mimics that I-72 switch? Just imagine a treatment that could trick a highly pathogenic virus inside a human lung into thinking it's back in the bat cave.
Forcing it to bind to restrictive proteins instead.
Exactly. Intentionally driving it into a harmless stalemate. We started today looking at a landscape the area is incredibly murky, this muddy water of bat reservoirs and human pandemics. But through the lens of a microscopic fishing trip, we can actually see the specific single molecule switches at play.
It really is incredible what we can see now.
It is, and it really makes you wonder what other viral double agents are just waiting to be discovered. Keep exploring, keep questioning, and thank you for joining us on this deep dive.